AJ Roberts, and I'm a research scholar with Reasons to Believe. My area of expertise is molecular biology or virology, and I'm really excited to be back on RTB Live. I'm glad you're joining us today. I'm joined by a very special guest, a friend of mine, Mark Clark. Mark is a professor emeritus in political science and national security at California State San Bernardino. Mark is also one of the visiting scholars here at Reasons to Believe, part of our scholar community, and you may have seen some of his blogs before. He's blogged for our scholar community. He's also co-authored a book with Ken Samples and Hugh Ross called Lights in the Sky and Little Green Men. And Mark has done a lot of work on Soviet propaganda and conspiracies along those lines. So I'm really glad to have Mark joining me today. I'm coming to you from my relatively isolated office in Covina, California. Mark's coming to you from somewhere in Colorado, and we're both doing pretty good. But in this time of COVID and everybody wearing masks, Mark and I both have a little bit of underlying respiratory asthma issues. And so I don't know about Mark, but I get a lot of looks when I walk around coughing from time to time. And so hopefully won't have to take a break from the live stream to cough some. But if I do, don't worry about it. It really is just an asthma flare. So, hey, Mark, why don't you say hi to everybody?
Hi, how are you guys? It's good to be on with you, AJ. It's great to see you. I am coming from Durango, Colorado. I escaped California about 2.5 weeks ago to come here and recuperate in relative isolation, but with a lot of outdoors and streams and fly fishing and things like that. So it's a joy to be here with you guys.
We're really glad to have you with us, Mark. Really good. Really looking forward to hearing some of the things that you're going to share, and as we answer questions about conspiracy theories. But as we head into this, so Mark and I are going to sort of pass it back and forth. We're going to ask some questions, pull some of your questions in from time to time. But the roadmap ahead of us is we're going to start out with just sort of what are conspiracy theories and why do people believe them? How do they find traction? How do they get traction? And then we'll talk a little bit about virus origin for SARS-2. That's how we'll refer to the virus that's causing COVID. We'll call it SARS-2 or SARS coronavirus 2. In comparison to the first SARS outbreak, which will be SARS or SARS-1. And then there's another coronavirus called Middle Eastern Respiratory Syndrome virus, MERS for short. So those are our abbreviations. And we'll talk a little bit about the origins of those viruses and SARS-2 in the middle or second block. And then in the last block, we'll talk a little bit about how we think Christians can think more clearly about things that often lead to or contribute to conspiracy theories and how to sort of pick apart what's true and what's not and how we wish Christians would respond. And so we've got 3 blocks and Mark and I will be passing them back and forth. But we're actually going to start off with Mark and I'm going to let Mark tell you, Mark, what is a conspiracy theory and why do people believe them? How do they gain traction?
Sure. It's a good question, AJ. I watched a video recently by a detective, a retired detective, who talked about conspiracies. And, you know, in the criminal world, they have conspiracies. In the real world, we have real conspiracies. Conspiracies are legally defined as when 2 people or more agree to participate in an illegal or destabilizing act and then carry out some action. It cannot just be to agree, but it's actually to carry out an action. And conspiracies do exist. I mean, you know, criminal conspiracies exist, political conspiracies have existed. Nixon did, in fact, you know, conspire to cover up the Watergate tapes. So they gain traction, though. The larger conspiracy theories tend to gain traction because people need answers. They want answers in times of confusion. Yeah. And so, you know, it gives you a sense of personal agency if you think you have some kind of control over something that's fairly incomprehensible, fairly confused. And so that's the reason why. But there's some underlying, it's interesting because I know that's the next question you're going to ask, AJ, so I'll go ahead and anticipate it. But there's some underlying research by psychologists that talks about not only what is the thinking patterns, the belief systems of conspiracists, those who generate conspiracy theories, but the times and conditions under which conspiracy theories are more likely to be accepted. And typically they found the 2 most common things is under conditions of stress and anxiety. Which this pandemic. Go ahead.
So under stress and anxiety, is everyone more likely to believe a conspiracy?
That's a good question. I think it's, I'm not sure the research says that. It shows that more people are likely to buy into conspiracy theories. I mean, one fact is literally 50% of Americans believe in one conspiracy theory or another, at
Wow.
least one. And so it's a fairly widespread thing. We saw that too in our book, Lights in the Sky and Little Green Men. About 50% of Americans will believe in UFO phenomena or know someone that believes in it. So those two conditions, stress and anxiety, bring about the greater opportunity for people to buy into conspiracy theories. But then they notice something else. It usually occurs in an information or media environment that is suggestive of conspiracies. And that's the environment we live in today. There's so much polarization politically in this country and in the world. And then you have a very active campaign of disinformation and propaganda launched by at least 3 countries. The State Department has identified China, Russia, and Iran are putting out a tremendous amount of disinformation. Carnegie Mellon found out, in fact, did research and said that literally 50% of the tweets we're seeing now on conspiracy theories are generated by bots. That is, they're automated. And so there's a huge amount of confusion and things like that going on in the world. So
Hey, Mark,
that's what we're seeing.
So you just used the word disinformation. I mean, yeah, that's different than misinformation, right?
Correct. Misinformation is conveying inaccurate facts. Disinformation is the intentional effort to convey bad information in a way that would be harmful. And so that's what we saw. The Soviets, the KGB was really good at disinformation. Or they actually have a word for it, dezinformatsiya in Russian. So you've heard of that term, I'm sure.
Хорошо.
Хорошо. Молодец. So so disinformation is purposeful, intentional misinformation designed to deceive, confuse, or detract from the truth. And that's what we have in the media environment today.
Thanks for that. Yeah.
Sure.
So what kind of conspiracy theories are we seeing with SARS?
We're seeing, there's a new Wikipedia page, not that I use that as my first and primary source, but it's an interesting source. If you go to Wikipedia, type in COVID-19 conspiracies, the pages are endless, and it's being constantly updated with references to articles. There's probably 100 conspiracy theories, but the more extreme ones we're seeing are that China has launched a biological weapon. This is a biological weapon created by China, intentionally designed to hurt the West or a lot of the world. That's one. Bill Gates is the other conspiracist. Yeah, he's out there trying to make money off the vaccine. So he started, and that's actually prevalent in particularly Africa. In the Middle East, we see a whole range of new conspiracies that the Israelis have created this, either intended to hurt Muslims or, in fact, to sterilize them once they get the vaccine. So there's a variety of conspiracy theories. Those tend to be the more outrageous.
And I guess it depends on where you live on which conspiracy theories gain the most traction, right? Because I guess in China, maybe there's some disinformation that America started this somehow.
Well, we do. That's part of in fact, the State Department report, AP, Associated Press did a report on the State Department's report that suggests that the 3 are coordinating their disinformation campaigns to blame it on the United States. That somehow Olympic Games that we participated in for our Army in Wuhan in October or something like that generated this.
Okay, so maybe answer this question, Mark. Do you have a favorite conspiracy theory, or do you have a favorite SARS-2 conspiracy theory?
The one that I'm most interested in is something I think you're going to address. Was it a deliberate, intentional, created virus? Did the Chinese and that Institute of Virology intend to create a virus? Take a sample from nature and give it some added functionality to make it capable of being a bioweapon. Now, I say that because we were accused of that all throughout the Cold War by Soviet propaganda again, especially the U.S. United Army's Military Medical Research Institute of Infectious Diseases was always blamed for AIDS, Ebola, you name it.
AIDS was a big one. The U.S. got a lot of flack, a lot of conspiracy around. It was created by the U.S. Army. So,
absolutely.
Yeah. So we've got a couple of questions. Mr. Jetty asks what the percentage of conspiracy theories are that turn out to be true. Do you have any idea what that might be?
I think it's extraordinarily small.
Yeah.
I think I would say in the range of 1% to 2%. But But, I think it depends on when I mean, one of the things that I found is, particularly dealing with people from different regions, is that um, I actually had a very good Syrian doctor friend, emigrated to the US, great guy, loved him, but he couldn't shake conspiracy theories about the Jews in Israel. It's just sad. Is it true? No, I've worked with a lot of Jews. They do not rule the world. I've just
Yeah.
not seen that. So
Someone named Tina Chang asks, how can you know if a person is receiving disinformation? How do you know if what you're reading is disinformation? I think that's a great question because, really, it's sort of like, okay, you in the intelligence business, how do you pick apart what's likely to be true and what's probably disinformation?
yeah, that's a great question. I learned it. I spent a lot of time researching as a grad student at the Naval Postgraduate School at the Hoover Institution. I went to USC, but I did research in the summer for what we called kind of the Soviet perceptions management. So I spent a lot of time reading defector testimonies, literature like that. And it's kind of like getting a PhD. You somehow stumble onto figuring it out. I do not know of a particular method per se. But what I strongly recommend is that you look at the information, you ask the question, if this is true, what's the inference? If it's not true, what's the inference? Does someone have to gain from this?
Yeah,
What is the source? And RT, for example, RT used to be called Russia Today, is a news agency that really works for the Russian government. That's a source you probably want to realize is, even when it employs American journalists, and by the way, they're hiring a ton of journalists
yeah,
because a lot of they're out of work.
yeah.
But they tend to employ state-sponsored effects. So you have to kind of study a variety of things to really get a handle on that. But that's a great question.
Yeah, so I think this is maybe along the same lines. There's a question by someone named SP Loden or Sploden. I'm not quite sure how to pronounce it, but it's a more technical question. He asks if you've used Bayesian reasoning to effectively avoid believing and propagating likely false information. If you
That's
have,
a,
you might want to explain that for the layperson.
yeah. Well, I haven't, and I'll
Okay.
tell you why. I think there's a simpler approach that we're going to talk about in the third section. And I think, I mean, that's what they use Bayesian reasoning for. I mean, and at least in my field, is to determine, for example, a submarine crashed at sea and it sank. We don't know where. And they use Bayesian reasoning to understand the water currents, the direction, the temperature, and they start narrowing the search area down from several tens of thousands of square miles to several hundreds, maybe several tens of square miles. That's a very much more complex form of reasoning. But I think for today, I want to offer a more modest approach that I think most people can gather and grapple with, and it might help our viewers watch it and understand it a little bit better.
Yeah, great. So we kind of know what a conspiracy theory is now. We know what disinformation is. We know a little bit about how to begin to ascertain whether what we're reading might be biased in a way to intentionally mislead us, and so it would fall into that disinformation category. But I've certainly encountered a lot of people out there who are writing blogs and or posting videos, and they'll make some certain conclusions, and their conclusions are really, from my vantage point, it's not that they don't necessarily have a bias or they're leaning towards one explanation of like how this all started or how it got out of control, uh, but it's that they really just don't understand some of the finer nuances, and so I would consider that, and you would too, I think, misinformation. And they're not sharing something inaccurate intentionally to mislead us, but they are being inaccurate in what they share, perhaps because they don't know more of the details, or perhaps none of us yet know the details, right? There's so much that's still changing about the situation. That's one thing we all have to keep in mind is we are learning as we go, and the more information we get always requires us to reassess.
And that'll be good. We'll do that in a little bit. I think it's great. But AJ, I want to throw a conspiracy theory out to you that you can then maybe turn into something a little bit better. One conspiracy theory has gained a lot of traction in the United Kingdom is that 5G towers, the cellular infrastructure for the Internet of Things, is causing coronavirus, COVID-19.
Yeah.
What are viruses? How do they
Yeah.
change over time?
Yeah, that's just such a crazy theory. The first time I heard it, I thought, "Oh my word, people just really don't understand what viruses are and what viruses do." And So viruses they're non-living. I say they're non-living because they require a living cell in order to make more viruses. They need the cell's machinery and resources and energy, but they're non-living biological organisms, if you will. And they're really pretty simple. I mean, even the really big viruses compared to the smallest viruses, they have three basic components in common. They have a nucleic acid, the genetic information like DNA or RNA that can be in a couple of different forms of RNA or DNA. It can be one piece or many pieces or circular or linear, but basically it has nucleic acid, and then it has proteins. And the proteins will help protect the nucleic acid. The proteins will also uh, mediate how a virus enters a cell. And so you may have seen pictures of the coronavirus, which are like balls with spikes coming out of it. Those spikes are proteins that are used to attach to cells and initiate virus entry. And so all viruses will have a nucleic acid or a type of nucleic acid. All viruses will have proteins. And then some viruses, a lot of them will have a third component, and SARS-2 does. It'll have a lipid membrane that coats the outside of the virus that those spike proteins are embedded in. And those membranes, for almost all the viruses that have membranes, are derived from the host cell membrane. And so when a virus infects the cell, as the virus is leaving the cell, the newly produced virus is leaving the cell, it picks up a host cell derived membrane. And so one of the other things that a virus has to do to infect a cell is not just attach, but those membranes have to fuse with the cellular membranes. And that's also something that the spike protein for SARS-2 does. And so really to think that 5G technology in any way could create a biological entity like a virus is just really bizarre. I just don't think that people understand what viruses are. I heard a slight variation of that. The 5G technology was damaging our immune systems somehow and making us more susceptible to SARS-2 or to the actual disease, the COVID aspect of the disease. And that's some conspiracy theories or all conspiracy theories, perhaps. It takes a very short amount of time to sort of throw out a rogue or a bizarre idea. And to try and start to address it really takes a lot of time because you have to pick apart, sort of, well, okay, what is a virus? Could 5G technology cause that? But when you take the flip side of that theory, that somehow it's damaging our immune system, then it becomes much more difficult to reason your way to a negative response or a negative answer, like, no, that's not likely. And that's because the immune system is so complex, right? And because different types of irradiation, like UV light or even chemicals that can cause mutagenesis in your DNA, changes to your DNA, those types of changes happen in our immune system, and they can happen through sort of natural exposure to things in nature, and they can also happen to artificial exposure to things, chemicals that we create, pollutants or things to that effect. And once you start talking about mutations of the immune system, then it becomes much more complex. But there's certainly no positive evidence whatsoever that 5G technology is damaging our immune systems in any way whatsoever. And I would think that would be highly unlikely. But because it's such a complex thought that they've woven into their conspiracy theory, it actually would take months to start to pick apart, well, we need to look at this, we need to look at this, we need to look at this, but there's no positive evidence to support it. And I think that's really important to maintain and understand as we keep talking about conspiracy theories and data and sources and origins. What's the positive evidence? It's not just what evidence is missing, but what evidence is there that actually leads me to believe that might be true.
Okay, that's good. So you've talked about the components. What's the difference between SARS-1 MERS and SARS-2?
Oh yeah, so those viruses I mentioned all in the front. So there's a whole bunch of different kinds of viruses. There are viruses that can infect bacteria that really have no impact on human biology at all. There are viruses that infect plants. And then there are all kinds of viruses, respiratory viruses, that infect us and other animals as well. And usually the ones that infect us don't infect other animals. But sometimes a virus that infects, say, a dog or a cat or a bat can make a species jump. It can jump out of the species out of the cat into a different species. And so that's one of the theories behind where SARS-2 came from, is that it came directly into the human population through bats. And one of the reasons that we think this is if we look historically at the first SARS, which occurred in 2002, we know that that virus and many, many other viruses, completely different from coronaviruses, we know many viruses are harbored in bats. Bats carry lots of different kinds of viruses in nature. And so in nature, SARS, the original in 2002, went from a bat into an animal called a palm civet, or a masked palm civet, or a civet cat. It's sort of a weasel ferret-like looking animal. And then from that animal, it most likely went immediately into the human population. And one of the ways that we know that about the first SARS virus in 2002 is because we were able to do sequence analysis of the viruses that we isolated out of humans, and we were able to do sequence analysis of viruses isolated out of bats and out of palm civets or civets. And we compared those sequences, and we asked how similar are the sequences. And we saw that the human isolates compared to the civet isolates were 99.5% identical at the nucleotide level, which is the RNA level, right? Not the amino acid level, but even more sensitive identity at the amino acid level. And so it was that first original SARS was also pretty similar to some of the viruses that came directly out of bats. If we look at MERS, we know that MERS came from bats through camels into the human population. And again, we know that because if we compare sequences of human viruses to camel viruses, we find virus sequences that are as high as 99.8% identical.
Wow.
And one of the other ways we can tell for sure that it came from civets in the case of SARS and dromedary camels in the case of MERS, so we can actually isolate virus from civets. We can isolate a virus from the camels, and we can test their blood, their sera, the same way we get blood tested or serology tested for antibodies, which means we've been infected with the virus, but we've cleared the virus. And so there were civets that tested positive for antibodies to SARS. There are camels that test positive for antibodies to MERS. And so MERS first emerged in 2012, but it still gets introduced into the human population periodically. There's already been dozens of cases this year alone in 2020 of MERS being passed from camels to humans. Now, MERS is really pretty scary. It's got about a 35% case fatality rate, with elderly people much more susceptible, like they are to SARS-2 and like they were to SARS. But we don't have a pandemic of MERS because it doesn't spread easily from human to human. And so that's really what's caused this to be a pandemic, is that it spread rapidly through the human population. But the question becomes, well, did it originate in animals as well? And if you've been following this at all, you know that one of the primary theories is that, yes, it absolutely, almost certainly originated in bats. And in fact, there's one bat isolate that's pretty well known right now in the literature and by people in the media because of its high degree of sequence identity with SARS-2. And that's 96.5% identical. And so that's pretty high, but it's not as high as it was for the camel for MERS, right? Camel isolates to human isolates were over 99% identical. And the civet to human isolates comparison in the original SARS outbreak also over 99% identical. And so this is pretty low in comparison if you compare the bat virus to the human virus, 96.5%. And so that leads many scientists who are studying the sequences and studying how coronaviruses change, and they've been doing these types of studies for 4 or 5 decades. It leads them to speculate that there's probably an intermediate animal host that we haven't identified. Now, we don't know that for certain, but sort of a Bayesian type reasoning, right? We make that inference that it's probable that there's an intermediate that we haven't identified yet. Some people speculate it's something called a pangolin, which talk about weird animals. A pangolin is really strange. It looks sort of like a scaly anteater, and it's from Malaysia. So over 2,000 miles away from Wuhan. And yet those animals are sold in various markets in the Wuhan and Hubei area. And so they speculate that maybe it came through pangolins. If you do a sequence comparison to pangolins across the whole genome, across the whole 30,000 bases of RNA, the percent identity is not very high, but in some areas, it's very high, almost 100% identical to pangolin sequence. In other areas, not so high, maybe only 80% identical. And so that leads to this other bit of really technical information that people who study coronaviruses and many people who study viruses in general know this about coronaviruses. They do something called, they go through a process called recombination. And what that means is you can take bits and pieces of the genome, and proteins are modular and genomes are also modular. And what I mean by that is certain segments of proteins and or certain segments of genomes can sort of be swapped for each other, and they have enough similarity that they can function in a substitute way. Well, nature actually swaps bits of genetic information, recombines the genomes of coronaviruses on its own all the time. And that's in fact how we think SARS emerged in 2002. It's how we think MERS emerged in 2012. There were recombination events going on in nature. One of those recombination events allowed it to move from a bat into a civet or into a camel. And then it was able to go from the civet or the camel into the human population pretty easily. So there's all kinds of signs as we look at SARS-2 genome that recombination has taken place. But as you can imagine, if nature is swapping bits and pieces of the genome in and of itself in natural hosts, and you start to ask the question, was this maybe tinkered with by a human in a laboratory somewhere in the Wuhan Virology Institute or something like that? Well, it becomes a little bit more tricky to ask, well, maybe a human swapped some genetic information and nature didn't swap it. And so then you have to kind of start picking apart, well, are there other signs of human manipulation? And one of the things that you would expect is there would be a high degree of nucleic acid similarity or identity, again, if it was human manipulation, because you have to start with known sequence, manipulate it, and then introduce it into what you want. And so really knowing about how coronaviruses recombine and knowing about how they emerge historically in nature really leads me to a confident analysis that SARS-2 probably originated in nature. Now, can I really pick apart whether or not it definitely originated in nature? Well, for that, I think we need to think about how it might have entered through a trader or a vendor or through a laboratory worker. And so what do I mean by that? Well, the bats are sold in market, but the bats have to come from somewhere. And so there are people who will actually go out and trap and trade and sometimes even raise bats, and then they'll deliver them to the market. And so you can imagine that if the bats are harboring a virus like SARS-2, if it came straight from bats, which we're not certain it did, but if it did, if it's harboring a virus like SARS-2, then that trader could become infected. And that trader doesn't even actually need to take an infected bat to the market. Just an infected trader needs to appear in the market, trading perhaps other animals like pangolins or something else to that effect. And then that infected trader could pass it on to somebody else. The same is also true. These bats are harbored in the Virology Institute in Wuhan, and researchers use these bats to study coronaviruses in order to learn what we can about coronaviruses so that we can be prepared in case there are outbreaks like this. And so the question becomes, well, it could have passed from a trader or a vendor into the human population, or it could have passed from a laboratory worker into the human population. And how do we sort of discern which one of those two is most plausible or most likely? Well, I like to think of it this way. People who come in contact with bats in nature, or even vendors or traders or trappers of bats, they almost certainly aren't wearing a whole lot of what we call personal protective equipment. And yet, having worked on the original SARS at the NIH, we work with lots of protocols and procedures in place, including extensive personal protective equipment, which covers our face, our hands, our entire body. We breathe filtered air. We go in and out through containment facilities. And so So the question becomes, is it more likely to have passed from a bat into the human population in nature, or in a laboratory where there's personal protective equipment that's being implemented? One of the other things that you could think about is maybe it didn't come straight from a trader into the market, but the bats have to get to the Wuhan Virology Institute somehow. And so maybe it was in transport of providing bats to the laboratory that a vendor or a trader came in contact with an infected bat. And that person then later traveled to a market or someplace else. The problem with this outbreak in particular is that we don't know who the index case is. That's the person who was the first case. And so if we knew who the first case was, we'd be able to say, oh, it was a lab worker, or oh, it was a trader or a vendor. But we don't have that data. And we can't even necessarily guess at where or who that person might have been, especially because up to 40% of SARS-2 cases are asymptomatic. So the very first person could have been an asymptomatic case and spread it to somebody without ever knowing that they themselves were sick.
That brings up a question.
We may never know. We may never know if it originated in nature or in a lab.
Yeah, that brings up a question. Donald Carlson asked that. How hard do you think China is working to find patient one or the index case? I don't know that we'll know. Do you? Because
I don't think that we'll know. And you know, I think we're going to talk about this in this next segment here. But when we talk a little bit about distrust of governments, but I think that, you know, do we want them to find the index case? Yes. Do they want to find the index case? Maybe. Right. So, I mean, if they have information we don't have, then maybe they don't. But at this point, I have no reason to believe that a natural introduction from nature into the human population isn't completely plausible and reasonable. And it's actually my preferred explanation, because why blame someone for something you have no positive evidence to support, right? So
you know, it's interesting because Anthony Fauci, you know, everyone, either the bogeyman or the hero, depending on who you talk to nowadays. I listened to a podcast of his in October or September of 2018, and he was asked 2018. He was asked this point blank, and this is by a former. It's called the Intelligence Matters. It's a podcast. Former acting director of the CIA, Michael Morrell, asked him point blank, what's your biggest fear, a man-made virus, bioweapon, or a, you know, naturally evolved one? And he thought about it and he goes, well, I am concerned about the man-made ones, but we see so much in nature that's happening already, and we're encroaching further and further into what we've never been encroached upon, has never been encroached upon before. He said that is by far my biggest nightmare, what keeps me up. And that was a little over a year plus a few months before all this happened. I thought it was
it really makes me wonder what changed back in the early 2000s, right? Because these viruses have been in bats for decades, if not centuries, if not longer. And yet we started seeing a lot of bat viruses enter the human population since 2000. And some of them are coronaviruses. Some rabies comes from bat, Hendra, Nipah. These are a couple of other viruses that we know originate in bats. We think that Ebola virus originated in bats. So what happened with sort of the human bat population that started creating the opportunity for more introduction of these viruses out of bats into humans? But I'm with Dr. Fauci on that. I mean, we're constantly monitoring viruses that can jump from animals into humans. Influenza. Everybody's really, really uptight, scientists and virologists in particular, about the bird flu, right? H5N1. We thought that was going to be the next pandemic, and we were kind of surprised back in 2002 that wasn't an influenza virus, that it was the first of these pathogenic, like truly pathogenic viruses, coronaviruses for humans. We were surprised, but not too surprised.
Yeah. Well, I want to throw on, because you talked about how scientists think about it, and I know you.
Yeah, that's how scientists think about it. How do government
agencies? Well, I think it's fascinating. You know what the director of national intelligence put out on April 30th, a statement. It sides with the scientific consensus that it was not man-made. So that way, they don't have the resources to check that out. They're relying on the open source literature of all the science community. So they basically accepted that assessment. And then the two things they're looking at, the only two things they're comparing is, was it strictly by human to animal contact somehow in nature, or did it come from the lab? And the reason why it's fascinating, because there was an open source report about 2 months ago by, it's a company called the Sierra Group. They actually have a subdivision, MACE, that reported cell phone, they tracked cell phone data around the Wuhan Institute of Virology. And in October, there was a period of about 12 days in which there was a reduced amount of cell phone usage. And then they showed overhead imagery where you didn't see a lot of traffic going in and out. So this is satellite imagery of that area. So for a while, I thought that was what contributed to the DNI's kind of assessment vehicle. But it turns out there's more recent, 2 other open source groups, Bellingcat is one, and I forget the other, have both critiqued that open source assessment because they only relied on 7 cell phones, which there's any number of explanations. That's not many. It's not good data source, right? And then the overhead imagery they used, if you expanded out slightly, you'd see that there was traffic construction signs built around the Wuhan Institute during that time. And it was a period when there was a holiday season in China. So that indication, that suspicion that there was maybe something going on, that outbreak might have started then in October, which led to the infection spreading, at least from the open source stuff I've looked at, doesn't seem that reliable right now. So if they're still assessing it, that would be part of what they're doing. They're going to throw a lot of stuff at this, a lot of resources. So it'll be communications intercepts, signals intercept, measures and signatures. They're going to try to do human intelligence collection. They've certainly done social media collection, data. My guess is we don't have many really good human sources of contact and information that could be corroborated. Our human intelligence collection in China is very tough. It's a very closed society. So that's how they're approaching it. And they're literally doing what I'm going to explain a little bit later, is they're doing an analysis of competing hypotheses. And I'll talk about that in a minute. But it's fascinating what they're doing, and let's wait to see what's coming up. There's some questions out here that I wanted to throw at you, if that's okay, unless you had something to add right away.
No. Let's go to some of these questions. I see one here from Kirby Hansen, and he wants to know what's the take on peer-reviewed papers, which have technical merit and hypothesize that COVID-19 was created in a lab. Well, I spent a little bit of time talking about part of that, but at a slightly more detailed level without getting too technical on anyone. As we compare those sequences, some of the speculation that's been in some very low-tiered journals, and what I mean by that, it's not a rigorous peer review, and maybe it went through peer review. It's questionable as to whether or not it did, but it actually did get published. And so some of those have speculated that there are viral sequences from HIV and SIV, which is the monkey equivalent of HIV, that are present in the SARS-2 spike sequence. And so I actually, with a couple of other scientists, have taken a very close look at some of those sequences. And what we found is that there's not a high degree of identity between those 6 small segments that were reported in this paper, that were reported as HIV sequences in the spike sequence of SARS-2. There's really not a high degree of identity. They're spaced out. Some of them are in reverse complement order, which means they're backwards. If you were to read one strand of the RNA, it's not reading as message, it's reading as complement to message. And then also some speculation over something called a furin cleavage site, which is the technical term for a site in the spike sequence where a protease, that's a protein that cuts other proteins in two. It's a cleavage site. So it's where a protease cuts another protein in two. It's where it cuts the spike protein in two. And so spike has 2 domains, spike 1, spike 2. It has to be cut in 2. And that's what starts that fusion event between the virus membrane and the cell membrane. And so we looked at the furin cleavage site sequence, which is unique to SARS-2 in respect to it's not present in the bat virus that's closely related to it, and it's not present in the pangolin sequence that's closely related to it. So it's unique to SARS-2 in comparison to those potential origins, and yet it's not unique to SARS-2 in the respect that SARS-2 is a coronavirus with a furin cleavage site at the boundary of the spike 1 and spike 2 domains. There are other coronaviruses, including MERS and some of the ones that cause upper respiratory and cold-like symptoms in humans that have furin cleavage sites in that same place. And then there are some that don't. So like SARS, the original, another virus called 229E, and another one called HKU1. Those are ones that cause a cold and upper respiratory. Also coronaviruses, all in the same family that don't have the furin cleavage site. So what you can do is you can go in and you can say, well, look at this specific sequence. If this specific sequence from SARS-2 really came from HIV or SIV or was constructed in a laboratory, then we would expect to find it perhaps in isolates that are used in a laboratory environment. Or conversely, we wouldn't expect to see those sequences someplace else in nature that just kind of suggests that those sequences happen fairly randomly. And so one of the things we saw with the SIV HIV sequences is they aren't specific in the right orientation. Some are in reverse sequence. They aren't 100% identity matches. They're not close together. They're not in the fusion domain. They're in the opposite side of the spike protein. And when we look for those sequences elsewhere in nature, we actually find those sequences in organisms like Dungeness crab and a couple of other organisms that are totally unrelated to viruses at all. And so those sequences aren't very specific. And so it's not a smoking gun. And where they appear in the spike protein sequence doesn't make rational sense if I were trying to construct a spike protein that might be more lethal or more pathogenic or better at attaching to the human receptor. The same with the furin cleavage site. If we look at the specific sequence from SARS-2 and we ask, do we see this anyplace else in nature? The answer is yes, we do. And we even see it some places in nature with the flanking sequences, not just the cleavage sequence. We find it in something called easy for me to say, a Mycobacterium phage. And we also see it in domestic barley. And so these speculations, these claims that there's positive evidence or there's evidence that we can construe there may have been human tinkering, I would say, au contraire. No, there's not. Not only is there a lack of evidence we would expect to see, but what they're claiming is positive evidence isn't really positive evidence because we see it in totally unrelated organisms.
And I would think too, this just kind of basically gets to my question: is it created as a biological weapon? It's not the most pathogenic virus, SARS-2.
It's not.
And there's other models that would be better to follow to make it far more dangerous to human population.
That's absolutely right. If we were taking everything we knew about coronaviruses and trying to create a more pathogenic coronavirus, we would have made other changes. These changes don't fit into the models that we have as how to increase pathogenicity, with the exception of the furin cleavage site. But again, the furin cleavage site's not necessary for the coronavirus to be pathogenic because SARS, the original, didn't have a furin cleavage site. It had a different type of protease cleavage site that allowed its spike protein to be cut. And it is thought to have a higher case fatality rate globally than this one does, although that's still to be seen. So
yeah. Okay. Do you want to move to the third section since we're
let's move on to our third section so that we have more time for Q&A. So I do want to make, I guess before we move on to the third section, I just want to comment from a shout out to somebody on my Facebook page. And as I just sort of answer one more time, will we ever have definitive proof if it was in nature, if it arose from nature transmission or a laboratory transmission? Well, yeah. I mean, if we come up with somebody's laboratory book that documents that they had an identical sequence, or if we isolate the virus in a natural reservoir, something like an intermediate host, like a camel, then we can get more data that can push us down one of these forks of which is it. But for now, I just want to say that about a week ago, a guy named Mark Peterson posted on my Facebook page in a long, lengthy exchange on these topics. He says, AJ, I don't believe there was any conspiracy to release the virus. I believe the virus originally came from bats and then was taken to the virology lab in Wuhan to be studied. This lab's been cited for sloppy protocols, and I believe the virus accidentally escaped, most likely when an infected worker left the lab didn't follow proper safety procedures, and then spread it to other people in the population. He said, that the Chinese government in Wuhan basically realized what was happening. They ultimately initiated draconian measures, and they put Wuhan in lockdown. They stopped movement to the rest of China, but they let people leave Wuhan. And so he's really sort of questioning the underlying motives of the Chinese government in containing it, and that they basically sort of dropped the ball, and they let it spread. And so my response to Mark on my Facebook page was, Mark, it's plausible. There's nothing unreasonable, unrational in the way that you fit the data together. And yet, because of primarily, as someone who's worked in one of these high safety laboratories, and I know the protocols that we need to put in place, and because I have numerous observations of how this virus has been transmitted naturally from people who don't have any protective equipment or protocols, I just find it more plausible and more likely that it came from nature than from the laboratory. But Mark, there's nothing wrong with the way that you've reasoned through this, and which one of us is ultimately right, we don't know the answer to that yet.
Yeah.
So Mark, as we move on into the third segment, this is really sort of a Christian response segment that both you and I will weigh in on. But I think that, even from the title of our RTB Live today and the way that I pitched off the ways that we want to be thinking clearly, I guess that's really, I'll emphasize the other one too, love boldly. As we think about those things, how do you think about this in light of how we as Christians should think about various conspiracy theories, and specifically those related to COVID?
Well, thank you. I think one of the things that I want to suggest to everyone is, first of all, you have to pay attention to this. Facts by themselves don't speak for themselves. It's the assumptions that we have, the beliefs, the theories, the kind of images we have of other states, other actors that really influence us. So as our text put this up for us, there's a question that I want us to be able to ask. Think about it this way. When I give you this statement, when you hear it, I want you to think of the first thing you get in your mind: a man lying in a gutter. Now, Nick will post it up, but if you think about it, any two people are going to interpret that differently. For example, a man lying in a gutter could mean he's a bum, or he's a homeless fellow. Or another person could say, well, a man lying in a gutter is someone that needs help, or he's drunk. I did this with a friend on a hike one time, and he said, oh, he must be murdered, because he's been reading murder mysteries. What's interesting is that simple fact of a man lying in a gutter doesn't explain what's going on. It's the assumptions that we have that allow us to make inferences. And in the comment you had from Mark Peterson, I saw a lot of assumptions built into his question that were good. So I just want us to be aware, we're going to take the same data, but it's the assumptions. Frequently, they're implicit, they're not explicit, and they influence how we make inferences. One of the problems I have with vast majority of conspiracy theories, maybe not all, is that they regard their assumptions as what we call axiomatic. That is, they are unchallengeable. And that leads me to believe that they're engaging in what we call a hypothesis confirming bias. They stumbled upon a hypothesis that they think explains the data, but it's a series of assumptions that may be driving their assessment, their inferences, more than the data. But then they tend to ignore, downplay, or refute contrary evidence. So one of the ways, the way in which I would suggest for Christians to think about it is through a process of abductive reasoning, not inductive or deductive, but abductive. One of the things that scholars—
Reasoning to the best explanation?
Correct, yes, reasoning to the best explanation. And the way in which I'll kind of explore it here is it's a technique of intelligence analysis. It's called an analysis of competing hypotheses. It's a subsection of, or a subset of abductive reasoning. What they found is the traditional way in which we analyze things can lead to cognitive biases. One of the traditional ways we do is we come up on the first satisfying assumption or hypothesis, and then we seek evidence to explain it or defend it. But then it takes time before you can get on. One of the problems with that is it's called satisficing. It's a strategy that only looks for minimal amounts of reasoning, and then it can produce that hypothesis confirming bias. So analysis of competing hypotheses is you take each hypothesis or each theory and treat it as an assumption or as a hypothesis to be tested. And you have to evaluate each hypothesis with confirming, disconfirming, and ambiguous evidence. And what you find is over time, it takes a little time to do this, but it's a more modest intellectual approach, I think. And it's one that says, at the end of the day, it's not the one with the most evidence in favor of it. It's the one with the least disconfirming evidence against it. That tends to be the more reasonable one to be explored. And that's what the intelligence community is doing with those two, that assessment that I talked about earlier. They are going to use an analysis of competing hypotheses. They brainstormed this early on. They did a hypothesis constructing brainstorming session. They explored every conceivable one, and they narrowed it down to those two. And now they're putting all their resources out to try to find out what evidence least disconfirms the one that they're dealing with. And it's a harder, so intellectually, I think more modest. And I think for Christians, the thing that I would encourage them to think about in this way is, if it helps us avoid our cognitive biases of satisficing, seeking the first one. But two, one of the more damaging things that can happen is if we buy into a conspiracy theory, it can destroy our credibility. And I want to give you one example. J. Werner Wallace, a retired detective, talked about it. He talked about his experience when he was first a detective and he went to a crime scene. And there he was with three senior detectives with him. And when he looked at it, he said, "Oh, this is what happened." And it turned out it wasn't. And he lost all credibility with those three senior detectives. Until they retired out, he did not have credibility. And I think the Christian witness to the truth claims of the gospel is the same thing. Are we, by buying into or latching onto a conspiracy theory that can be shown to be wrong or shown to be irrational by other people, do we lose our credibility with the truth claims of the gospel? I mean, that is our first priority. That is our first priority. Matthew 28, right?
Absolutely.
So I think it's a more modest intellectual approach. I think it's one that Christians can do. It's going to take a little training, and I'll probably do some blog pieces on it a little bit later on how to think about it and how to do analysis of competing hypotheses, not the complex way. But you can type it into Google search and you'll find a whole bunch of information on how to do that, what they call the ACH. So that was my way. AJ, what's your way? How would you like to frame the question?
Yeah, I think I really appreciate the way that you frame it, Mark. But I think, again, I'm just going to come back to the think clearly, love boldly, pray always. But what I'd really like each person to do when they're trying to ascertain with what they're reading, if it's disinformation or if it's hype or if it's somebody speaking more out of emotions or maybe somebody that has a distrust in one of those categories that you talked about before. I think that's the first thing that you need to start with as you're looking at something, is to ask yourself first, what is the source of whatever it is I'm engaged with? Is it an article? Is it a blog post? Is it a Facebook post? Is it a video that's traveling around social media? Ask yourself, what is the source, and what do I know about the source? Does the source have some expertise in this field? Who writes the source's paychecks? Right? Mm-hmm. Are they a scientist who's sort of in public health, sort of in the forefront of global emerging diseases? Is it a public health official, for some local government or national government? Or is it somebody that sort of is a biological weapons watchdog? Or is it somebody who works for RT, like you mentioned earlier, Russia Today? Ask yourself, who's the source? Who writes their paychecks? And whether or not they might have a conflict of interest in getting people to think about things a particular way. I put a couple of really good articles up on my Facebook page again today, and both of them do a really good job of sort of assessing, is there sort of an underlying agenda, which is one of the first questions you can ask when you're looking at a particular source. Yeah. The second thing is almost as important, I think. So pause, ask yourself that first question. The second question is, what is my bias? What am I tempted to want to believe? and to recognize, oh yeah, I distrust the government on the whole many times over, and so maybe I'm going to be more tempted to believe those stories or accounts that put the government in a particularly bad light. Maybe I don't distrust the US government, maybe I just extremely distrust the Chinese government. So anything that puts the Chinese government in a bad light. Ask yourself that question. Become aware of your own sort of heart bias and cognitive bias. And then ask the question that I mentioned a little bit earlier when I said these two things are so important in the way I look at which one I find more plausible. What is the positive evidence supporting a particular conclusion? This is actual evidence. This evidence actually leads me to think that this explanation is more plausible. What evidence is missing? And if the evidence is missing, should it be there, right? If somebody actually constructed this in a laboratory, there probably will be definitive evidence that I should be able to find at a nucleic acid level. Is that evidence there? If it's not, it's missing. And so what does that tell me? That tells me maybe I don't want to believe that one until I have positive evidence, right? So, and then
I'm gonna join you on that though, because you said something that's fantastic. What am I inclined to believe? You know, I studied national security. I have a high distrust of China. I've written on articles on back in the 90s, should we contain China? And I argued back then, no, not then. But I'm aware of my bias. And so that's absolutely crucial because I've seen it with questions that I've been getting. You know, China, because it violates treaties, it would automatically do this. Well, there are reasons why it would not launch a biological weapon.
Right.
A tremendous number of reasons why they would want to avoid it, because you can't target individual people. It always comes back.
Right. And they could get infected with their own virus.
Absolutely. Right? Yeah. So anyway, so I wanted to support you on that because I think we're running time, but that's absolutely one of the most important things to take away.
Yeah. And again, just to reiterate what you just said, that would be my fourth point as well. You know, what are the possible explanations? Sort of weigh out the different explanations and ask, you know, can I get somebody I trust? Tell me, rank them in likelihood. You know, which one of these seems most plausible, most likely in light of everything that we know. We do have some time for some Q&A, and we're going to do that. And then we're going to also take a lot more questions in the RTB Live Extra, which will be coming up next. Mark and I will go ahead and answer all your questions that you have out there, as many as we can, in the RTB Live Extra. And then that podcast will be available to you on Saturday, this coming Saturday. You can go out there and hear the rest of the Q&As. And we have lots of other questions from Dylan and from Nina and from Jessica and John. And we just think that those are going to fit best in the RTB Live Extra segment because of the topics of your questions and what you're asking. But I just want to say one more thing as Christians, as we think about this and as we converse with others who are sometimes fearful and stressed, and sometimes we're fearful and stressed. I've heard somebody even go as far as to say that not a Christian, but a non-Christian, sort of criticizing engagement with COVID on a global level. Like, this just has to be evidence that God doesn't exist, right? They're sort of taking this and turning it into positive evidence against the existence of God, and they're really questioning God's goodness. You know, I've talked and I've written on this topic a little bit. My AMP talk last year, that's one of the conferences that RTB hosts every year. I talked about God's purpose is revealed through science and suffering, and so I sort of addressed that, like, why do bad things happen if God is so good? But in regard to this, I'm going to come back to something that you said earlier about Tony Fauci and what he was really worried about. Was he more worried about a bioweapon, or was he more worried about nature being able to produce something like this? And not to sort of, I mean, it is going to end on a sort of a sobering note for us, but not to try and get everybody down. But I don't look at this as positive evidence against God's existence. I actually think of it in this way, that we've been expecting, we being virologists and global public health officials have been expecting a really bad pandemic to emerge for the last several decades. And we thought it was going to be influenza, and we still are very worried about some of the influenza strains out there. And as we've been wrestling with trying to contain COVID and SARS-2, we've realized that we're not as well prepared as we needed to be. Some of our stockpiles in the U.S. weren't replenished. We don't have the healthcare capacity that we need to deal with a coronavirus pandemic on top of a very difficult flu season that can quickly overwhelm our medical system the same way that Northern Italy's medical system was overwhelmed. And so I look at this and I think, this could be a lot worse. The case fatality rate could be a lot higher, like MERS, or it could be a lot higher, like what we anticipate for H5N1 influenza. And it's not. In some places, the case fatality rate is less than 1%. It's never as low as the flu annual mortality rate, but in some places it's very low. In other places, it's considerably higher, but it could be much higher. And so I kind of look at this as maybe it's God's mercy getting us as a global society to sort of wake up and say, oh my goodness, this is bad, but it could have been worse. What do we need to do to prepare for future possible pandemics? And I think that we really do need to be focusing on that. And maybe if we're focusing on that and not so much on conspiracy theories, something deeply good might really come out of this.
So I would add, it's interesting why people often buy into conspiracy theories is because they want some personal agency in a confusing, chaotic, helter-skelter kind of environment. I mean, I think personally, I like to think of taking agency by the gospel, following Jesus Christ. And then secondarily, subordinate to that, how do we take agency with this? How can we help prepare our friends, our family, the healthcare system to be better prepared for future pandemics that most likely will occur, as you pointed out, and as others did too. So I think that's an absolutely great way to think about it. So thank you for that. I've told you before, you impacted my wife and me with that assessment before, so I'm glad you brought it up again. Thank you.
Yeah. So we may circle around and repeat that again before we take off, but sure. We're going to tackle a few questions here, and I think I'd like to see if you've got a response to Elliot Larson's question, Mark. He says a recent paper in the Gospel Coalition and a dialogue between Tim Keller and Francis Collins, who, by the way, just won the Templeton Prize.
He did.
Congratulations, Dr. Collins. Spoke about Christians being more likely to accept conspiracy theories. I guess the assumption is more likely than non-Christians. Is this true? And what is the evidence? Nice. I like the last question there. What is the evidence?
Yeah. Yeah. It's interesting. In the one that I watched with this detective, J. Warner Wallace, he had the same comment that Christians seem to be more likely to accept conspiracy theories. And he made an argument. I don't have evidence. I haven't done the research on that. I'm kind of curious now. That's data in search of a good hypothesis for me, right? So I have to find the data, and I've got the hypothesis. I'm going to go find the data. But he made an argument that I thought was interesting, Jay Werner, because he didn't become a Christian because of rationally looking through all the different arguments and comparing and then saying, okay, this has got to be it. People do that, but the vast majority of people become Christians for not irrational, but non-rational means, either through, tragedy in home life or becoming aware of their finitude or the awareness of God in nature, or 100 different reasons that have little to do with fact finding and evidence gathering. It's only after they become Christians that some will go on to do studies like what we're doing here, like apologetics and things. Again, I don't know that that's the definitive answer, but I think it's a great question, and I really appreciate that. I think that's something that, that's a good blog piece or 2 or 10 down the road.
Yeah, and maybe to fuel or put some fodder on that for your consideration as you think about how you're going to blog about it and break it up. It's an interesting question, because if we circle back around to sort of, how you laid out, what is it that allows conspiracy theories to sort of gain traction, and why are certain people attracted, or when are people attracted to conspiracy theories? You said it was under times of stress and fear or anxiety, right? And so I guess, maybe that's one thing to think about, Mark and Joel, or whoever asked that question.
Elliot.
Are Christians, can Christians at times tend to be more fearful or anxious? And unfortunately, I think the answer to that question is yes, but I think it's because we get our eyes off of Jesus, right? The author and perfecter of our faith. We take our eyes off of God is involved and God's heart towards us is good. And so I mentioned this in my last RTB live, or at least in the extra bit, I can't remember which one, that,
Mhm.
I meditate on Psalm 139 because Psalm 139 just speaks to every aspect of my life and how intimately God knows me and how much God loves me. And I look at that and I find great reassurance in that, and that God has numbered my days. Not only is he intimately acquainted with every action, every word before I do it or say it, but he's numbered my days since before they were born until the very last one that I lived, before I even drew my first breath. And even beyond that, through eternity, uh, my eternal state is secure in Christ. I'm going to live with God forever because I've been born again of the Spirit. And so I find great peace in that. And I think that if we can focus on God and God's word and God's truth and God's plan and God's sovereignty, and not get so caught up in the different narratives or the stories or the accusations or the rhetoric or the fear around us, then I think we're going to be less likely to believe conspiracy theories because we're alleviating the underlying fear and anxiety and stress.
I'm going to add something to that because I've just written a piece on how Daniel may have coped with the trauma and captivity. It just came out, I think, 2 weeks ago. And one of the things I find fascinating is Daniel was a young man when Babylon came calling and pretty much knocked out Pharaoh Necho of Egypt. Judah lost its allies, and all of a sudden they were ransacked. The temple, a certain level of people. He was taken on forced march 900 miles to serve a pagan god and king. His name was changed. He was stripped from his family, his Jewish religion, his ethnic identity. He was likely castrated, and yet he exemplifies a life of hope and joy at a very crucial time. And it's because I believe he literally took agency by looking at Isaiah 49:6. It is too small a thing for you to be my servant, to become a light to the Gentiles and to restore the tribes of Jacob. And that's what he does throughout his time there. He exudes this quiet confidence in conditions, and he believes in the sovereignty of God under conditions that are far worse than we're experiencing under this pandemic. He's literally got no choice. He's stripped from everything, and yet he finds a way. And I recommend everyone go read the book of Daniel and look at that as a starting point as we move forward.
That's great. So we've got your Bible studies lined up for you for the next week or two. You got Psalm 139 and the Book of Daniel. Yeah, we've got a good friend Brian that wants to know if Batman would be immune to SARS too, and I think the obvious answer to that, well, is he's part bat, and so probably, because these viruses don't seem to cause disease in bats, and in fact maybe they even serve an ecologically good purpose in bats or for bats that we don't know of yet, because we haven't done those types of research. We're getting some questions about sort of how long I think this is going to last, and how do we re-enter society safely, and what about the drugs, what about the vaccines, and I'd really like to answer all of those, I think, in the RTB live section, or extra section. But there's one question here by John Pinto that asks if I could speak to the gain of function work that was being done in the US and China on the bat viruses that might have accidentally been released from the lab in Wuhan. And yeah, so I actually posted again on my Facebook page, and I really encourage you all to go out there and interact with me. That information should be here available on the YouTube live stream for you to be able to access that link to my Facebook page and my Twitter page. And this is also a question that Dylan Carr asks. He asks, where do I go to for reliable sources? And so, whenever I post to Facebook or Twitter, I almost always post the supporting evidence from scientific peer-reviewed literature or from the sources in science who I know personally and or I know through personal networks, and I trust the work that's coming out of their laboratory or out of their group. And so I post those sorts of things there. But on my Facebook page, in regard to gain of function, I posted a very good article, and I made this point that people who think that gain of function equates to work that's more like bioweapons, trying to make a virus more pathogenic or more targeted towards humans or an agricultural target or whatever, that's misunderstanding entirely what gain of function experiments actually are. And what gain of function experiments actually are is they're trying to pick apart the biological things that we observe in nature that are already there and ask, what is it about that virus that makes it now able to infect a bat cell and a human cell, right? And so what is it that changed so that when we go out into nature, like we do for influenza,
Mhm.
and we surveil, we collect viruses from waterfowl, and we look at those viruses at a molecular level in the laboratory, and we ask what's going on in the influenza virus population in waterfowl in Asia, and we're monitoring it, we're surveilling it, because we sort of have these sensors, these biological sensors out there, what's going on in nature around us. And when we see certain things happen because of studies we've done in the lab where we can pick apart gain of function, uh, we can identify, oh, there's a potential situation brewing here and we need to be careful, right? Or, oh, this entire flock of chickens
Mhm.
has become infected with a highly pathogenic virus for humans. Let's go kill all those chickens before it gets out of the chickens and gets into people, right? So to think that gain of function studies, and this is misinformation or disinformation from people who have put it out there. Either they don't understand what gain of function is, or they're intentionally trying to deceive you or make you believe something about these experiments that are going on in these laboratories as something nefarious. They're just misrepresenting what gain of function studies are.
Looks like we have a couple more questions. There's one I wanted to ask. It was good. Judy Stallings asks, do you feel that conspiracy theories arise out of the brain's desire to create story and the resulting release of endorphins when story, accurate or inaccurate, is created? You know, that's a
Wow,
great question.
that's a great question.
It is. Steve Willings, another visiting scholar, sent me an article today that I haven't had time to read, but the assessment basically was why people buy into conspiracy theories. It gives them a sense of meaning and purpose, agency. But the story or the research article he gave to me, and I wish I had read it fully, had time to, basically says there's no proof that long term this satisfies all those needs that we have. So we have this need. Is there endorphins? I don't know. I'm going to have to read the article, but it's a fascinating question. I thought it was great that she asked that question.
Yeah, Judy, that is a great question, and I love. I mean, this is one of the things that I actually get the most excited about is I believe absolutely that we are people of story, right? And we want story, we need story, we are embedded in story. We are part of the greatest story ever told, and the story is all about us, but all about us and our relationship to God, and therefore it's really all about Jesus, the bridge between us and God. And and I think that that's true no matter where we're looking in the world around us, whether it's social interactions or conspiracy theories or even at scientific discovery levels. We are people of story, embedded in story, for the purpose of story. And so I think that that's really hitting on something. And yeah, man, come out on Facebook and interact with me. I want to know who you are and what led you to ask that great question.
That's a great question. Yeah. Um, wow.
So I think we have time for maybe one or two
Okay.
more questions. Let's see. We could answer one of Daniel Carr's questions. I think Daniel asked, where do I go for information? And, you know, uh, I often will go to a source called Stat News. It's an online source that has, for a very long time, done extensive research into emerging viral diseases, emerging bacterial diseases, infections, sort of global public health. But they do it at an extremely responsible level. And occasionally there'll be a little bit. You may have heard of Stat News. I think they leaked one of the stories about one of the vaccines before it had gone to publication or before the information was released by the organization, the biopharmaceutical company or the FDA. And so they kind of got a little bit of flack for sort of releasing information because it caused a jump in the stock. And so, I mean, not everyone is subject to criticism when they misbehave. But for the most part, I trust Stat News as a source for the science, right? I wouldn't necessarily follow them for business stock advice or something like that. But and then another person that I follow is Scott Gottlieb, who used to be the former director of the FDA. I follow him on Twitter, and he's got great group of followers. And sometimes when Scott says something that's not particularly accurate, the people who post comments to his tweets sort of like jump all over him like a rabid pack of dogs. And so that's good. Got checks and balances out there on social media. And then there's another guy named Trevor Bedford, who is a researcher who's actually studied the changes in the viral sequences in the human isolates from Wuhan isolates to Washington State and he collaborates with many, many other people. And there are tens of thousands of SARS-2 sequences out there now, which is just so phenomenal. This time in the first SARS outbreak, we were just getting the first full-length sequence of the SARS coronavirus. And now we have tens of thousands of sequences of various isolates. And so we're able to sort of track how the virus is mutating. Most of the mutation rate seems to be pretty slow, and none of the mutations seem to be correlated with higher degrees of disease or lesser degrees of disease. We don't see a lot of mutations that are suggesting yet to us, based on the limited experimental data that we have, if it's going to be able to evade vaccines or drugs, or if there's more than one strain out there. The answers to all of those are everything that we know suggests that if we have a drug, if we have a vaccine, it should be broadly applicable. It should probably be durable in the sense that it won't need to be renewed like influenza. Influenza undergoes major changes antigenically because it does something other than recombine. It does something called reassorts because its genome's in pieces. And so anyway, so I go to Trevor Bedford at Fred Hutchison, who does a lot of his information. He'll either post on his lab's website or he'll tweet about it on Twitter. And he often will cite other people's work from Johns Hopkins health security researchers and public health officials. I find John Hopkins a very good and reliable source. We're talking on conspiracy theories. And so always in conspiracy theories, I'm always a little bit sensitive to some people distrust the Chinese government. Some people distrust all government. Some people distrust our government too. And so to point people to government sites, I still think is a credible place to find out what's going on globally. The World Health Organization, again, sure, there's some room for scrutiny and criticism from time to time, but they're doing an excellent job in a very difficult situation, in my opinion. And as far as the U.S. government sites go, the Center for Disease Control. And yes, there are conspiracies out there that are painting the CDC in a bad light, and Bill Gates and the Gates Foundation. And again, decades in virus research and aware of public health threats. These are organizations that I deeply respect and I trust them for the most part. But we're a community of watchdogs in and of ourselves. And so when one of us screws up in some organization, there are usually many of us there to sort of not so lovingly correct them and say, hey, what the heck's going on? But yeah, so that's where I go.
And I would just add, for many of you don't trust newspapers and things like that. What I do is I take several different newspapers and look for the common facts that they're speaking about, verified facts. And I try to separate out where the opinions, the spin, the political agenda is, and take those facts and then go analyze those on my own, because that's so much more important. But there's really good, even if you don't like the New York Times, has some really good reporting and good editorial control. Not always, and it makes mistakes. So does Washington Post, but Washington Times, there's others, Wall Street Journal. Der Spiegel, go to foreign sources. If you really want some really interesting takes, is go to foreign sources. Australia has some really good research that comes out of their newspapers. Der Spiegel is very good. It has a good timeline on the coronavirus in China. They did some research. They actually interviewed people in China before they were shut down and closed off from interviewing. But you have to be careful. You have to, as we talk about, understand the source, understand the motives, and then compare when they're speaking facts versus when they're speaking their opinions or their interpretation of those facts. And that takes a trained eye. You just have to spend time at it and talk to people that you trust to help you sort through that.
Super. So we've got a 4 more questions out here. I'm going to punt 2 to the live extra from Daniel and Leo. I will answer your questions in the live extra, but I'm going to address Joel's and Sploden's. And I know it's the second question from Sploden, but he asks, can different sequences result in functionally identical proteins? And the answer is yes. so that's an easy one to answer briefly. But basically, different nucleic acid sequences can result in functionally identical proteins because either they result in no amino acid changes, or the amino acid changes that result are what's called sort of I just blanked on the word, but basically the substitution at the amino acid level is a homologous type substitution, and so it doesn't, so like a basic amino acid for a basic amino acid, and so it doesn't alter the protein's structure or chemical capacity for function. And so yes, it's possible to have different nucleic acid sequences and still have functionally identical proteins. Okay, and then Joel's question is, if the virus isn't man-made, then why and how did it spread so fast across all the nations under different temperature conditions? And the answer to that is because viruses do that. And right, so the question that you ask is presupposing that there's a sensitivity of this virus to temperature and to humidity. And we know that it's not as sensitive to those two things as influenza is. And yet there are many other viruses that I could point to, like measles and smallpox and polio, which spread through many different countries, all different climates, all different temperatures, and persisted for a very long time. And so different viruses behave differently in different environments. Some viruses, like SARS-2, seems to be pretty hardy, and it seems to be able to persist in a variety of environmental conditions. Certainly true of measles, smallpox, polio, and many other viruses. So.
And I wanted to tag onto that because I saw a biologist that I work with at university posted a graph of the second stage or the second wave of the influenza, 1918 influenza. And the second wave was 3 to 4 times more powerfully destructive than the first wave. Yeah. I didn't research that historically. It looked accurate.
No, it is accurate. There were actually 3 waves of the first the 1918 influenza, yeah. And we only have isolates from wave 2, so we don't know how the virus changed from wave 1 to wave 2.
Interesting.
And you may hear people out there propagating misinformation that we're preparing for a second wave. Well, globally, we're still in the first wave, and so. Yeah. You know, we haven't gotten through the first wave of SARS-2.
No,
okay,
yes, thank you.
So Mark, thank you so much. I've loved having you on here, and I love the insight that you bring and the expertise that you brought to the discussion. Please join us for RTB Live Extra, and we look forward to seeing you there. And if not there, out on social media. Everybody, God bless. Have a great week.
Thank you.